and Ian Dewberry from South Africa. So welcome everybody to What's New in MPMI. This is the virtual seminar series of the MPMI Journal. We're happy to welcome you, excited to see people joining from around the world. That's one of the real pleasures of this, and also was the reason we started. So when we started this back in June 2020, I really felt that we needed some way to connect with colleagues around the world, and also to include colleagues from around the world in our discussion of our science. And that connection has been tremendous. I see many of you on every time we have one, and of course, always new people. So the idea is that we have all this great science happening in the journal. And an important thing here is to help you, the audience, to connect both with our science outside of the actual pages of the journal and also with our authors. And that's one of the greatest things, to hear the authors talk about their own work. And today we have Thomas Griebel, who will be talking to us about a very interesting story of cross-priming with abiotic stress. But before we get into it, I want to remind you that this will be recorded, and you can always come back to watch the recording. People don't need to register to watch it. And that you can also go to the same place and sign up for our next talk, which is Adam Huberger and Janak Joshi. And I think that is coming in May. But anyway, that's going to be another terrific talk. Pectobacterium virulence, wild potato. It's a great story. Thomas is talking about work that was from our last focus issue, which was from highlighting one of these top 10 questions in MPMI. What are the questions that drive our research? What are the big questions we're thinking about? And one of the really biggest was, how does the abiotic environment influence the biotic? And our next focus issue is actually coming out this month, and that's going to be on the endomembrane system and plant microbe interaction. So I can already see a lot of really great papers showing up in Just Published and in First Look, so definitely take a look. So before we start, I want to introduce Amelia Lovelace, who will be our co-host today. Amelia is one of the assistant features editors for MPMI. Do you want to introduce yourself for a moment, Amelia?
Yeah, sure thing. So again, my name is Amelia Lovelace. I am a postdoc at the Sainsbury Laboratory in the UK under Dr. Wemba Ma, and I mainly focus on bacteria. So I'm very happy to co-host and looking forward to hearing about Thomas's research.
Great. I should also point out that Amelia Lovelace is one of the organizers of a science communication concurrent session at the IS-MPMI Congress that's going to be happening in Providence, Rhode Island, I think in July this year. So that will be, that's happening right at the beginning. So definitely want to be on time for the Congress so you can catch that. It's going to be her and a number of the other assistant features editors. Somebody is asking about the transcript recording this. Yes. So actually, I'm glad somebody brought up the transcript, because what we try to do is to have somebody edit the transcript after the talks, and then we can post the edited transcript along with our our Zoom recording. So that would be tremendous help if somebody would like to volunteer. If you do, please type it into the chat, and we'll get your name. And this helps to increase inclusion, because many of our listeners, English is not the first language, and this is a helpful thing to have the transcript as well. It also improves accessibility for hard of hearing scientists around the world. And it's particularly important with a scientific talk, because Zoom transcript does great, but not so great with scientific words, and it does terribly with names. So I would really recommend anybody who would like to help. This is how you can improve make this seminar more inclusive. So thanks. Okay, so now on to our talk. We have one of the big questions that came out of our discussion at the last MPMI Congress was, what are the big questions that are driving us right now? And one of the top two questions, the second question was, how does the abiotic environment, the physical environment that surrounds plants and microbes, how does that influence the interaction between plants and microbes? And so many interesting papers that were contributed to that focus issue. And one that caught my eye right at the beginning was the one from Thomas Griebel that he's going to be telling us about today. A great instance of cross-priming. So we think a lot about priming of one biotic interaction affecting a subsequent interaction with the microbe. But this is cross-priming between an abiotic stress or reading the abiotic environment with a subsequent biotic interaction. So really interesting, and exactly the kind of questions that I think our research is going into. So very excited to have that today, to have Thomas here to share his work with you. And he might talk about one or two other additional things where this is all going. So I am going to turn it over now to Thomas Griebel, who is coming to us from the Dalum Center of Plant Sciences. Thomas, you can share your screen and get started.
So hello also to all of you from my side. My name is Thomas. I'm from the University of Berlin in the Dalum Center of Plant Science. It's a great pleasure for me that we got this kind invitation to present our publication here as a virtual seminar, and also thanks to Jean for the nice introduction. So we were very happy that we could contribute with our publication, Cold Exposure Memory Reduces Pathogen Susceptibility in Arabidopsis, based on a functional Plastid peroxidase system, that we could contribute to this focus issue about the role of the abiotic environment on interactions between plants and microbes. So let's get started. And I picked a figure from the same focus issue from a review from Omar and Kenichi Tsuda. And they nicely illustrate all the different abiotic challenges for plants in the environment. Temperature increases, temperature decreases, cold, freezing temperature, different light conditions, shade, UV light, with different water conditions, drought, rain, and flood. And plants, like all other organisms, experience such abiotic changes in the environment. And this even extends to biotic challenges. The outcome of plant pathogen interactions is affected by abiotic changes in the environment. These environmental variables, they can strengthen or weaken the dual plant pathogen interaction. On the one side, weaker or plant resistance or make it stronger, but on the other side also affect the pathogen by affecting its virulence, which might become stronger or weaker depending on environmental conditions. When combining different plant stress exposures in experiments, this can be, of course, done at the same time, and increasing complexity, which occurs at one moment, but you can also apply different stress treatments at different times. That's what we did. We used a kind of an experimental setup where two successive stress exposures were studied. You can apply one treatment, then have an interface or a recovery phase, and then apply the next treatment. And when you have such a successive stress exposure situation, of course, the first stress or the first treatment can be of disadvantage for the next stress experience. It could be without any effect, neutral, but it could also be beneficial for the second stress treatment. This is a concept which is defined in the concept of priming. Priming means that the first stress exposure, which is then defined as a priming exposure, has a beneficial effect for a later occurring stress treatment, stress exposure, which I call here the triggering stress. Both stress treatments are separated by a recovery phase, a memory phase, which allows, of course, reacclimation from the first stress, and also requires, on the other side, that the first stress is somehow memorized, and that there is a kind of a molecular memory. During the triggering stress, this priming can show different responses. For instance, the response in the second stress can be faster. It can occur earlier or be even more sensitive, or even as a stronger response, and thereby have a beneficial effect during the second stress exposure. So this is more or less the setting where our group started. Initially, we started in our lab the effect of cold exposure on a later occurring cold exposure treatment. So what was part of the setting is a cold treatment, 40 degrees at 40 degrees for 24 hours. You use in the lab Arabidopsis thaliana, 4-week-old. Then after the first stress exposure, we bring the plants back to the normal growth conditions, and several days later we apply the second stress exposure, initially another cold treatment at 40 degrees. You may wonder why we decided for a cold exposure. What is so interesting about the cold exposure? And I could give you many reasons and explanations for that. One really essential explanation for us as plant physiologists is that a sudden cold exposure creates an imbalance between the photosynthetic electron transport and the Kelvin-Benson cycle. And this is also a reason for our experimental setup, and that we apply the cold treatment, for instance, not in the dark phase, in the night phase, but 2.5 hours after the onset of light, we apply the cold exposure, and we use a full day-night cycle for 24 hours, so including a light phase and a dark phase to study the effect of cold exposures. So just to give you one example about some data we had before we started the project on pathogen response. And this was that we could show that cold priming reduces the expression of cold response genes during a second cold triggering. So at the end of a second cold triggering phase, several stress responsive genes, for instance, SAT10, which is responsive to chloroplast ROS production, showed a weaker activation when the plants were primed before the triggering stress compared to the plants which only experienced the second cold phase. And this is not the only gene. Several other genes showed the similar behavior, like here SAT10, SAT12, or SAT6, and we have a full transcriptomic data set published about such changes that we can observe during a second cold following a prior cold treatment. That was also more or less the start or the situation when I joined our plant physiology lab here in Berlin. And I came from the pathogen field, and I was immediately asking the questions, so what happens now to the plant pathogen response if there happened a prior cold treatment? And we addressed this question and applied Pseudomonas syringae tomato to our cold pre-treated plants. Although I guess it's not needed for this audience, I will give a short summary about the different layers of the plant immune system. So there is one layer where membrane-located receptors recognize so-called microbe-associated molecular patterns, or PAMs, MAMs, and this recognition activates a first layer of immune responses, summarized as MAM-triggered immunity. Virulent pathogens can overcome this. They use the type 3 secretion system to secrete effectors into the plants, and these effectors counteract some of these immune responses, increase the susceptibility of the plant, and the plant becomes susceptible, and the bacteria is it can overcome these resistance responses. When the plant or the plant cell can recognize some of those effectors and activate a second layer of defense, this is usually summarized as effector-triggered immunity and gives really a strong immune output and a strong immune response. Of course, today we also know that both the effector-triggered immunity and the PAM-triggered immunity, they are interconnected at several points. Okay, in our experimental setup, we used different Pseudomonas strains which work with these different layers of immunity. Of course, the virulent Pseudomonas strain, PST, here we see the situation in Columbia Arabidopsis thaliana plants for effector-triggered susceptibility. We can use a PST Hc- strain, which is deficient in a type 3 secretion system and cannot deliver type 3 secretion system dependent effectors into the plant cells. Or we can also use Pseudomonas strains which deliver effectors like PST-AVR-PM1 or PST-AVR-PS4, which deliver effectors which are recognized in Columbia Arabidopsis plants. Actually, so this was our initial experiment. We applied the code for 24 hours, waited 5 days, brought the plants back to the control conditions. Then we performed infiltration of the different PST strains, and 3 days later, we isolated the bacteria. And here you see the result and the outcome of this initial experiment. While for the PST Hc- or the ETI-inducing strains PST-AVR-PM1 and AVR-PS4, we did not observe a difference in bacterial numbers between control plants and prior cold exposed plants. However, in the experiment with PST, with the virulent strain, we could observe reduced numbers of bacteria after 3 days. So we concluded that cold priming reduces the plant susceptibility against PST. We thought of now continuing this, and were wondering what could be the effect of that. And a classical approach is take immune-compromised mutants and see which immune pathways are really affected. And one of this strategy was to use a null mutant of the plant immune regulator EDS1. Just as a summary, in the interaction with PST, EDS1, the plant immune regulator, contributes to salicylic acid formation in the plant. It contributes to defense gene regulation. This helps to reduce the susceptibility on the one side. On the other side, of course, contributes also to something which is called systemic acquired resistance. I will talk about that later. In addition, EDS1 is known to contribute to immunity mediated also by cell surface receptors and, of course, by intracellular TNPLR receptors. And we have the question is if there is a common cold priming mechanism which is functional in Colcio, but maybe also in the immune impaired EDS1 null mutant. So we repeated our experiment, used the wild type Columbia and the EDS1/2 mutant. And what we can see here is that we could confirm our initial results that cold primed Columbia plants showed reduced PST numbers compared to control plants. And in addition, also that the strong immune impaired EDS1/2 mutant also showed this effect and had reduced bacterial numbers when the plants were cold exposed several days before the cold treatment. To exclude that this is just an amplification from a lower bacterial starting inoculum, we also re-isolated 3 hours after infiltration, which we also did for all the other data sets shown here. And immediately 3 hours after infiltration, we cannot see this difference. So we really concluded that this is an effect which comes during the time of infection to limit the bacterial number stronger in the cold primed plants. We continued and changed our experimental setup. So what about these 5 days? What happens if we reduce this to only 1 hour? And our data showed that 1 hour was already sufficient. So after 1 hour after the 24-hour cold exposure, we more or less see the similar things. We see the reduction of bacterial numbers in Columbia and EDS1/2 lines. This was about the data we had when a nice publication came out 2019, and the colleagues Wu et al. They had a look at shorter cold exposures of only, for instance, 10 hours, and immediately infected the plants. And their data after a pre-exposure of 10 hours at 4 degrees in the dark, without a long recovery phase, before the PSD inoculation, they also observed similar things, that the cold pre-exposure resulted in reduced bacterial numbers. They didn't continue only with a 10-hour cold exposure. They did a nice variation of pre-exposure times. And in this variation of pre-exposure times from 1.5 hours to 36 hours, you can see that already 8 hours are sufficient for a significant reduction of PSD titers. And this continues until 24 hours. In the next experiment, the colleagues they did the opposite. They extended the recovery time from 1 hour, 2 hours, and 5 hours, and used the later infection. However, for this 10-hour exposure, cold exposure, they observed that already after between 5 hours and 12 hours after the cold exposure, the memory effect or the effect of the cold on PSD numbers is already gone. So there is only a transient effect when you only expose your plants for 12 hours in the dark. This is also quite a difference between both experimental settings. This is exposure in the dark, and we have exposure in the day and night phase. But this data set really helped us improving our understanding about the cold exposure. So we continued with our experiments. This is the data set I just showed you 2 slides ago. The effects 1 hour with an infection 1 hour after the cold priming and 5 days after cold priming. The picture looks quite similar. However, we think that the situation in the plant is quite different. So after 1 hour, there is still a lot going on from the previously experienced cold exposure, while at the 5 days later, most of this cold exposure changes in the plants are already gone. And we checked this by testing some cold responsive genes after the initial cold priming exposure, 3 days later, and also 5 days later. And what you see in this transcript analysis for Columbia, but also for EDS1, that of course, immediately after the cold, you see a strong cold response. However, already 3 days later, this cold response is nearly gone. And this continues also at 5 days. For the 5 days data set, our group has published 2 RNA-seq studies, which really show that 5 days after the cold exposure, the transcriptional state is pretty much similar to the wild type plants and only slightly affected, but all the initial cold response is gone. Okay, so how to continue? What could be the effect or what could cause the effect on the PST numbers after the cold exposure? One idea we had came from a priming scenario, which is quite familiar in the plant pathogen field. And this is the so-called systemic acquired resistance. And what happens during systemic acquired resistance, or how is that induced? In systemic acquired resistance experiments, you do primary infection of lower leaves. This induces, of course, defense responses. There is a signal cascade and some mobile signals going to the systemic uninfected tissue. And there already without a second infection, you can observe the defense activation at the metabolic or at the transcriptional level. So this is a priming scenario, plant pathogen, and we thought maybe this is quite similar to what we observed. Maybe cold priming induces a physiological status in the leaves, which is quite similar to the systemic acquired resistance induced leaf tissue. To get a first idea of that, we analyzed transcriptional data sets and we selected the gene ontology group for systemic acquired resistance. Here in this panel, you see the genes of this group from a SAR-induced state, and you see quite some variation in the transcripts when the leaves are SAR-induced. Next to it, there is the same set of genes immediately after cold regulation, where you observe slight changes already. And then we checked our 2 data sets 5 days after the recovery from the cold exposure. And this was very silent. And even if we saw some changes, these were usually unique to one of the RNA-seq experiments and was not confirmed by the other data set. As for instance, in the one data set, we saw that this pathogen is related to one gene was a bit slightly upregulated, but this was not confirmed by the other data set. So the transcriptional status in the cold-induced tissue in the memory situation seems to be not like the SAR-like status. However, as part of the definition of priming, it could be that some of those genes show a stronger or faster activation once the second stimulus, the infection, occurs. So we selected some of the candidate genes. For instance, the salicylic acid biosynthetic IS-1 or the salicylic acid responsive pathogenesis related to one gene. And we checked the expression after cold priming and during the infection with PSD. And you can see here that both genes, IS-1 and PR1, are definitely induced, as it is known in the orange panel. But in the cold prime tissue, it was not altered. It was more or less a similar level. So we didn't observe a faster or stronger induction of these candidate genes. Okay, again, we went to the literature and we found this really nice study from Singh et al. From the Laura Zimmerli lab. What they did differently to us, they didn't use a single cold treatment, they used repetitive cold treatments, and repetitive cold treatments, 1.5 hours per day, 4 degrees in the dark. And as soon as they applied 7 repetitive cold treatments, they observed what we observed, the reduction in PSD numbers. And quite interestingly, this effect also stayed for 5 days. So when they used a recovery phase or a phase between the cold exposure and the infection, they observed this effect as long as there were only 5 days in between, and it got lost after 7 days, which was really also confirming data from our cold priming, cold triggering studies. What they found that 7 repetitive cold treatments, after these 7 repetitive cold treatments, PTI responsive genes reacted stronger upon PTI activation. So they tested those genes after triggering with PST-HUX-C or flagyllin, and they observed a stronger activation of VERQ53, FRK1, or NHL10, and made a nice study how these PTI-induced genes are primed upon repetitive cold treatments. So we took that up and checked the PTI marker genes, NHL10, FRK1, and similar to the transcripts before, we did not really observe a difference between the control plants and the cold prime plants. So the stronger activation of the PTI responsive genes might occur after repetitive cold treatments, and but seems not to be an outcome of a single 24-hour cold exposure. Okay, we went back to our data from the cold group, also cold priming and cold triggering group. What they found, or what we found in these studies, was that a thylakoid-bound ascorbate peroxidase is a central regulator of cold priming responses during a second cold treatment. First of all, ascorbate peroxidases. What is this? Where do we find those? We can find those in many cell compartments. In the stroma of the chloroplasts, we have 2 isoforms usually, which is a thylakoid-bound ascorbate peroxidase and a free soluble stromal ascorbate peroxidase. Ascorbate peroxidases detoxify H2O2. They reduce H2O2 to water by using electrons from ascorbate. And there is this idea about the H2O2 protection in the chloroplasts, that the thylakoid-bound ascorbate peroxidase provides the first layer of detoxification directly at the thylakoids close to the photosystems. And the stromal version provides a second layer of H2O2 protection in the stroma. The post-cold regulation looks like this. So after cold exposure, TAPX transcripts increase slowly during these 5 days. On contrast, SRPX is very fast activated during the cold, but very fast goes back to control levels. We could confirm this in our experiments after the cold exposure, and we tested here the TRPX expression shown as ratio of the cold primed plants compared to the control plants. We see in Colombia and EDS1 this TRPX upregulation during the recovery phase. In contrast, SIPX, as expected, is strongly activated directly after the cold exposure, but back to control levels very fast. However, there was a bit of contribution for SIPX as this cold-induced induction seemed to be dependent on a functional EDS1. So this is just transcript data during the recovery phase. Of course, we wanted to have genetic evidence that Plastid ascorbic peroxidases contribute, and we used well-established knockout lines of TAPX and SIPX in our experimental setting. And these are the data. Again, in Colombia, we see the reduction in the cold primed bloods, the reduction of bacterial numbers after infection with PSD. In the TAPX knockout line, this reduction was very strongly reduced, and in the SIPX knockout line, it was nearly absent. So we concluded that our cold priming effect requires these Plastid ascorbic peroxidases. To get further evidence for this, we included conditional estradiol-inducible TAPX RNAi line. With this line, we can very specifically reduce the TAPX transcript levels, and we applied estradiol treatments during the memory phase, decided for 2 time points. And indeed, just as a confirmation, the TAPX transcript levels were reduced at the end of the recovery phase when we applied estradiol, and we continued with our Pseudomonas infections. So here are the results. Let's have a look first at the functionality of the line. The TAPX eRNI line without any estradiol treatments, just the control line and the cold primed line show a similar behavior as the Colombia line. So the cold priming reduced bacterial titers. Unfortunately, the MOX spraying already increased the susceptibility again in TAPX, but also in the Colombia line. However, estradiol specifically strongly increased the susceptibility in the TAPX line. So we concluded that the conditional TAPX expression after a cold exposure, that this increases the susceptibility against PSD. This brings me now to my summary. I try to make the summary very short. A 24-hour cold treatment reduces PSD susceptibility during infection, when the infection occurs immediately after the cold exposure, but also 5 days later. We could show that several components of the immune regulation were not affected, but we found evidence that Plastid peroxidases, SIPX and TAPX, that they contribute to this effect. We hypothesized that there is a chloroplast ROS or redox signal from the chloroplasts after cold priming, which then can reduce the plant susceptibility. Of course, we have many open questions. For example, is it really a common feature of SIPX or TAPX, or are there some specific SIPX or TAPX specific processes? What about compartment-specific ROS control in the context? We would also expect that there is a kind of cellular redox signaling from the cold primed plants via the cytoplasm, probably to the nucleus. When we talk about the chloroplast enzymes, of course, there comes the question about the photosynthetic stability. Although we did not observe effects on ETI or PTI-inducing strain, or like PST AVRPS4 or PSD-XC, in reducing bacterial numbers, this might not need to mean that there is not an increased resilience and a faster or better response, which does not show at the level of bacterial titers. It's also a possibility that this increase that there is an increased robustness of chloroplast effector targets, but these are all open questions. And just as at the end, I don't want to leave you without having shown no unpublished data. We had a look at the apoplastic ROS production. And it's well known that PTI triggers an apoplastic ROS production. We wanted to know how does it look like in cold primed Arabidopsis, also PAMs induce a plasma membrane located oxidase, RBOHD, which produces then specifically for the apoplast H2O2. And this can easily be studied in the lab, and that's what we did recently. And we see here in the Columbia line, for example, after the application of the PAMP NLP24, that we observed a slightly stronger apoplastic ROS response in the cold primed tissue compared to the control plants. We had a look at several mutants, also the knockout mutants of TRPX and SRPX. Interestingly, there was also an effect in the TRPX knockout line. We checked after flagellin 22 and NLP 24 triggering, and this ROS production was even slightly stronger in the TRPX knockout lines. So indicating that this is a response which is not affected by TRPX. However, we did not observe a difference between in the PTI triggered ROS response in control and cold primed plants in this SAPX knockout line. So with this, I would like to finish this talk today. I would like to thank, of course, all the co-authors, Margarete Bayer, the head in our plant physiology group at the University in Berlin, Dominik Schütte, a PhD in the lab, who's also currently continuing this project as part of his PhD. I would like to thank also two former members which contributed as co-authors, Alina Ebert and Hung Nguyen, both contributed to this project as part of their bachelor project and are now continuing their scientific careers in different labs and performing a PhD. Of course, I would like to thank all present and former members who contributed. I'm very thankful for the funding which came from the German Research Foundation for this project, and this funding took place within the Collaborative Research Center 973 of Priming and Memory. And of course, I would like to thank the Freie University for funding us and all the opportunities we have here in our labs. And last but not least, I would thank you all for your for attending this talk and for joining us also now for the discussion. Thanks a lot. And yeah, I'm looking forward to the discussions.
Thank you, Thomas, for a really interesting and insightful seminar. That was a lot of fun. I can see questions already, lots of little hand clapping there. Wasn't it a terrific seminar? And I just want to remind you that we already have a lot of questions showing up in the Q&A box. So if you would like us to answer yours, please type yours in the Q&A box. And I'm already seeing a few people typing into the chat that it was a really good seminar. So lots of enjoyment there. So I'm going to start off by reading them, and Amelia and I will take turns. So Piao Yang says, "Hi, Dr. Griebel, nice talk. I have two quick questions. One, how long could the cold priming persist?" And you did address that somewhat. And does it also work on necrotrophic fungal pathogens like botrytis cinerea? And two, so this is two questions in one, what are your thoughts on applying cold priming concept to agriculture? What challenges and chances are there? So lots packed into that one.
Okay, first question, very short, from our cold project, and we are really sure that the effect is goes away between 5 and 7 days after the prior cold exposure. That's what we have also already published, not on the pathogen question, but on the cold question. The necrotrophic question, does it also affect the growth of necrotrophic fungal pathogens like botrytis? We're right now testing. The experiments are running. What are your thoughts on applying the cold priming concept to agriculture? It depends. What I think it's definitely worth when this becomes more clearer, also in connection with energy saving, and for agriculture, when we think about greenhouses, does it, or there's a question, then would it benefit when we grow the plants in the greenhouses to reduce the temperature for a day, save energy, and improve their plant immune system? Or maybe we need to find out what the costs of this is. Maybe it's less biomass at the long term, but it might be beneficial, at least against different stressors. Yep.
So our next question is from Ludo, who asks about what inoculation method you used for the Pseudomonas. And I guess a follow-up question would be, do you think you'd see a similar response if you were to do a dip inoculation versus direct infiltration?
We used infiltration method within this data set, but we have also tested spray inoculation. And for Colombia, we get similar results. Yeah.
That's a good question. It's true that how we contribute the pathogen can sometimes have a big effect. So our next question is Bahir Basha Shaikh, who says, "Dear Dr. Griebel, very informative talk. Is it possible to apply this cold priming technique on Sclerotium rolfi causing stem rot of groundnut, a major crop in South India?" So I think everybody's wondering how widely do you think this might be a real thing? Your response.
I think this is a really good question, and this is really showing the perspectives also for such a crosstalk mechanism. Can it be transferred to other systems or even to crops? And is there a similar mechanism? I can what I can say. Yeah, we haven't studied anything else so far besides Arabidopsis, but yeah, worth testing.
Our next question is from Sventanka, and they ask two questions. Regarding the TAPX expression regulation under cold priming, is it influenced by light, and are they regulated by microRNAs? Very good question.
It's definitely that SAPX and TAPX show light-dependent expression patterns. It's already that you need, important to test the transcript regulation at similar date times, so they, because all these can affect the expression levels. And
microRNAs.
MicroRNA, I do not know if that has been studied so far in the APX ascorbate peroxidase field. There are different components which regulate APX expressions. Might be definitely also an option. Yeah, I don't know.
So our next question by Ryan Houston really touches on this question of light again, which is of course one of the biggest inputs for plants and something they're constantly sensing. And they're wondering about the timing whether and when light is required for cold resistance, because you did talk about how you were very careful at the time of day that you applied it and to make sure it covered a light period. So I wonder if you could just discuss that a bit.
This is definitely a hypothesis we have, that it needs the light phase during the cold priming exposure. But we haven't done experiments completely in the dark, or we also have the hypothesis that it really needs the drop of temperature during the light phase to really get the cold sensing in the chloroplast based on this imbalance between photosynthetic electron transport and Kelvin-Benson cycle. But this is our expectations. We haven't done experimental testing. For us, it was interesting. This is also why I showed data sets of colleagues that as soon as you perform the cold exposure only in the dark, you have different components of regulation. So this really makes a difference.
It makes sense. You're going to have different kinds of regulation of the photosynthetic apparatus, et cetera, in light and dark. And it would be interesting to see how that plays out.
Definitely.
I think we'll skip the next one about fungal pathogens because we've already discussed that. So Amelia, do you want to go to the next one?
Sure. So the next question is from Andrew, and he asks whether host plant immunity you saw that host plant immunity was unaffected by cold. So would it be more accurate to say that cold treatment makes Arabidopsis more susceptible to this virulence strategy of Pseudomonas syringae in particular, rather than to pathogens more generally?
Both is possible. It is possible that the virulence strategy of PST is weakened by the cold priming in the plants. At this moment, we can't really give an answer to that. It's definitely an alternative option, that it's a reduced virulence of the pathogen.
I'm going to
It is,
because
yeah, go ahead.
I was going to follow up because Pseudomonas syringae does have some effectors that target chloroplasts specifically, so maybe that could be tied in, and maybe those particular effectors are effective.
And on one slide, I mentioned a recent publication from our lab from the last year, also from the cold priming, cold triggering group, and they found that during the cold memory response, there is a change in components of the photosynthetic machinery. And I could really, if one of these components would be an effector target, then I wouldn't be surprised if this effector target would be more robust or less active. Yeah.
Well, that would be a fascinating possibility. I love the idea of effectors targeting the chloroplast and zipping in to block that kind of cold priming. Really interesting. Lots of good directions to go. Okay, our next question is by Henrik Stotz. Hi, Henrik. How about 2-cis peroxyredoxins and redox signaling for defense?
Yeah. Hello, Henrik. So we have initially also included the cis peroxyredoxins in our analysis. And as far as I remember from the first screening, we also had an effect in 2 cis peroxyredoxin mutants. Generally, when I did not talk so much about the unpublished data set, but what I can tell to this question is, as soon as we use mutants which are factored in redox signaling in the cells, in the cytoplasm or in the plastid, our effect is altered. So we really think that there's a redox signaling process involved.
Our next question is from Marta, and they ask if you have tried other abiotic stresses to prime against pseudomonas intolerance, such as nutrient deficiency.
We haven't gone for nutrient deficiency so far, but we have definitely some one or two projects in the lab running currently, which are also looking at other abiotic changes.
So
And that really is the question: how much crosstalk is there between the different abiotic stresses?
Yeah. So this is also in context of memory. Does this really matter if it has been a cold? Or does the plant maybe just remember, okay, there was a stress situation, I need to do some changes in my physiology? This is, I think this is really an interesting question, and we're looking forward to check or get the final data also from some other abiotic treatments in the same experimental setting.
Yes, I mean, there's also so much overlap between cold and drought stress, so many different directions. Okay, let's see what is next here. Ilya Stahl says, thanks for the nice talk, Thomas. Chloroplasts are important hubs for the production of specialized metabolites and their precursors, which possess antimicrobial activity. Any changes after cold exposure? Although SA doesn't seem to be changed.
So we have tested several components before the triggering. So at the end of the recovery phase, after these 5 days, there we didn't see a strong change in salicylic acid, jasmonic acid, or ABA. I think that's what we tested. During the infection, we only have the evidence from the transcriptional stages so far of that biosynthetic genes. But definitely a possibility. Yeah.
So our next question is from Lenka, who asks, who says, thank you for the interesting talk. They asked if you have any comparison with the effect from a longer exposure to cold, like 3 days instead of 1 day.
So within our research consortia, there were further groups involved with specifically also studied the effect of 3 days exposure to cold. So first of all, we haven't tested if this affects the plant pathogen response so far. What we observed that for longer cold exposure, also the regulations of memory changes. So with longer cold exposure, we our colleagues observed epigenetic modifications where we so far do not have any indication for our very limited 24-hour cold exposure. But no results, no idea for the pathogen response.
So complex, all these interactions. Our next question is by Nicolas Cecchini. Nice talk. Did you check vitamin C levels in the APX silenced plants? This is back to the metabolites. The Foyer lab showed a relation between VTC1 mutants and defense. And do you think you may be affecting calcium levels in plastids in primed plants, thinking of PTI, ETI connections?
Definitely the ascorbate levels are checked currently in the lab. I'm not aware of the results yet, but this is coming. And calcium levels in the plastids, I have no idea. Can't comment on that.
Our next question is from Eshla Deva, and they ask that they say that you mentioned that cold priming for 24 hours in the dark was done, and was done 2.5 hours after the light cycle started. How crucial is that timing?
So for our experimental setting, our cold exposure was not performed in the dark. So we just changed the temperature, and we changed the temperature. So we had have two different growth chambers, our normal growth chamber and the 40 degrees growth chamber, which transferred the plants to the 40 degrees chamber with the same settings, except a different temperature, but also with a full day and night light cycle.
Okay. And their follow-up was whether anyone's looking at doing the cold priming at different times of the day and whether that affects the memory at all.
Yeah, first of all, we haven't done this experiment, but my speculation would be, yes, it makes a difference. Maybe it does not make a difference at the level of bacterial titers. Maybe both can reduce bacterial titers, but I would speculate that it makes a difference at the level of how of the mechanism and the regulation, that this really differs, and the chloroplast component might be stronger when the cold priming, cold exposure starts during the day in the light phase than when it starts in the dark phase. And sometimes I even think, to get a bit to the environment in reality, a cold night is nothing which comes so surprisingly to plants, at least in the area where I live, where we quite often have cold nights, and memorizing each cold night is maybe not as important as a sudden cold period might be. Yeah.
Exactly. And then during the day, as you pointed out, when all this photosynthetic electron transfer is happening, and so it's much more susceptible, and all that's really turned off very rapidly in the dark. So I wonder if you'd see a difference between 2.5 hours after start of day and 2.5 hours before the end of day. I would imagine you would. I
would also imagine, I don't know about cold, but many years ago I did similar experiments with plant infection, independence of the daytime, and it was a difference when I infected in the morning compared to infections in the evening or in the night. And I would also expect that from a cold exposure. Lots of intersection there. Okay, George Lazarovitz, is the resistance sufficient to protect the plant? And he points out that the levels of bacteria would still result in plant death, but that probably has a lot to do with the amount that you inoculate with, right? Yeah, so for our bacterial growth assays, we use a very low infection or inoculation. And for this, we see symptoms, but definitely even with the PSD infection, our plants survive. Yeah, this would be different when you go for higher doses. However, when we go for higher doses, the leaf tissue for Arabidopsis collapses so fast, it's hard to see if there would be a difference for a cold priming response.
Our next question is from Jummer, and they ask about pseudostring and whether we looked at the transcriptome of the bacteria during this cold priming and what did they experience?
Definitely it could be that a cold prime plant, or the question is if the pathogen recognizes that this plant was cold exposed before. Of course, this is an advantage of a sequential and successive stress treatment. So the cold exposure itself, it does not, the pathogen never notices because it happened several days earlier. However, of course, it could be possible. I don't know if for such a virulent or such a pathogen or such a microbe, a cold prime plant feels different. But then it would also mean that there are components or signals, maybe through the apoplast exported, or that the leaf surface even changes after the cold priming exposure. Open questions.
Right. And of course, you're adding your inoculum, not under cold conditions, right?
Yes.
But
So
kind of
they're
after the cold.
exactly.
So we never see the effect of cold on them. Okay, the next is by Dr. Almonifi, who asks, are you planning to test combination cold priming with other priming factors like biostimulants on Arabidopsis susceptibility? So basically combining a abiotic and biotic pre-treatment.
Actually, this is one of the main project currently of Dominik Schütte in our lab. He is testing the question if cold priming can be additive to systemic acquired resistance. So it's a cold priming and a pathogen priming. Can this result in an additive effect, or is there some complementation? Yeah. But we haven't tested any further biostimulants, no.
Our next question is from Luda, and they ask, from an evolutionary perspective, why do you think plants have evolved to increase immune responses after being exposed to cold?
That's a very good question, and this is also a question which is really in the center of this. Coming back to this top two questions from the MPMI community, also the evolution of such processes. Definitely, on one side, it could just be a crosstalk and a side effect. On the other side, when we look at infection, when it occurs, when maybe not for pathogens, but for fungus-like situations, quite often you reduce a bit the temperature, you reduce or increase humidity. So there are several components about abiotic facts which improve the infection, and maybe such a cold situation could also be something like a warning situation. Okay, be careful, it gets a bit cold, maybe there comes an infection. But it could also be just a random crosstalk, activating things where the plants benefits in both situations. But yeah, the final answer I cannot give, and I can only speculate.
So kind of like a generic set of stress responsive genes, that there will be this kind of shared set of genes that will just be pulled out no matter what the stress.
This was very nice in the Sing et al. Study, which I showed from 2014 from the Laura Zimmerli lab. And they really applied very different stressors, and they always observed the same effect. So here it didn't even matter what kind of stress it was. So it was just, okay, be careful, something is changing, I need to be more resistant.
Right, there will be some specific stress responses, but the first thing to kick into play is just probably something affecting reactive oxygen species, et cetera, for that first signal. One thing that occurred to me with this evolutionary question is your Columbia is a line. Now, I'm not a Arabidopsis researcher, so you can feel free to correct me, but it's a line that comes from, well, I mean, it's collected in Columbia, but it almost certainly came from Arabidopsis populations that were used to cold, right? That lived in temperate parts of the world where there were cold cycles. I wonder what would be if you looked at plants that do not live in environments where they would expect, as you said, daily cold every night, you know? And I know there are some Arabidopsis populations from the Azores, or anyway, places where it really doesn't, you know, it doesn't get cold. I wonder what their response would be to that 4-degree treatment.
Actually, when we look at different accessions, we get a full picture of more or less everything. We tested several accessions in the lab. I can identify accessions which do not benefit, where it's more or less similar accessions, which even become more susceptible after the cold priming. So far, with the limited number of accessions we tested, we cannot make a conclusion if this is connected with the climatic conditions from the place of origin, where they were harvested or growing. No idea, but I definitely see a full picture of responses.
Fascinating. Really interesting.
Incredible.
I think we're going to stop there because our last question in the Q&A is something we've already discussed. But really interesting set of ideas and questions. And I love that fact that, Arabidopsis is not just Arabidopsis. You have all the different isolates who are carrying, have some local adaptation and carrying in their genome these either epigenetic or genetic changes that affect their responses. And that's certainly going to be, it's a whole can of worms there. See how that might interact. Anyway, this has been a lot of fun. Thanks for everybody who stayed through all the questions. It was a terrific discussion and set of questions. I'd like to thank, I'd like to thank you, Thomas, for sharing your research with us. And I want to point out to people who are still online that, if you read the paper, his email address is there, and authors always welcome comments and questions from readers. So that's another way to interact. So anyway, thank you so much, Thomas. Thank you, Amelia, for co-hosting.
Also from my side, thanks, Amelia.
Okay, goodbye, and we'll see you all in 2 months.